Alright, so here's my little video narrative for the protein gel electrophoresis that we did in cell molecular biology lab. To start off here, we have our setup. Typical to what we have been doing, we've been using these little holders here for the microcentrifuge tubes, as you can see, and these are actually the ones we do have in class. So we had set up a designated 4 tubes, one for each of the samples of the cell pellets, as well as one for each of the supernatant. And then there was a 5th tube provided that contained the ladder as well as the buffer mixture. Each of these samples was then set up. It had an equal part, which ended up being 20 microliters of the sample as well as 20 microliters of the buffer, and then they were centrifuged and collected. In the meantime, here's our apparatus. The gel was set up. There was a razor blade used right here that can cut off 2 of the sections along the sides that are more of like a guard, and you can see here the combs are already taken out of the gel wells. They're actually in the solution right here. The lid's set up. You have our battery that you can adjust the voltage and amps with, as well as the time. Then this lid was actually placed on top to the matching positive here and negative here section to apply the voltage to the solution. And here's our picture of the gel in the solution. You can kind of see to the fill line, it's on the other side, but there's 2 to 4 gels, and we did do 4 gels. Here we have the ladder sample in one, and then moving across, these are both cell pellet samples. 5, we did not use the well because it was broken, and then 4 and 6 were both supernatant. You can kind of see bands starting to form along the bottom here. And then we have a video here. This is the actual gel in the solution. You can see a little bit of bubbling going on, not too much, and that continuously bubbled until the 30-minute period is up, which we did 200 volts for 30 minutes. And then we took the gels out of the actual wells themselves. After that, you can see kind of here they were placed in a plastic tray and they were put on a shaker, which you can see here. This is actually the destaining process. So once the stain was washed off, they were allowed to soak around for about another 5 to 10 minutes to completely rid of the stain on the actual gel itself. And then for our result, I have a little bit of a comparison here. The marker, which is what our case was the ladder, you can see kind of like a few bands that are starting to pull down along the gel. They kind of run across. That's meant to be that way. It's meant to kind of strain out and get thinner as it goes down. For our samples, they all stayed at relatively the same area. It looks like the supernatants both had a singular band that was a little bit lower. It's kind of hard to tell because there was no UV light used, which does help illuminate the bands a little bit better. But from this data, you can kind of, well, I guess from this observation, you can kind of make the assumption based on this chart here that the proteins had a long DNA strand throughout them, throughout the DNA sequence in the yeast and the yeast plus sugar. It does make sense that they would have the same proteins, being that they are the same species, even though they are in different environments. So although the strands from the gel did not go down further, that doesn't mean it's necessarily wrong. It just means that the proteins were more complex. And then the supernatants having that extra band may have just been because it wasn't as concentrated DNA. And I believe that if we would do it again and run a little bit lower voltage for longer time, being that we have it, we would see more of like a fine separation. So that was our setup and results for the gel electrophoresis. And I believe that it was useful to determine the lengths of the actual DNA sequences that we had. Thank you.